Hello, my name is Betsy Espinal, and this is my final project for Genomics in the virtual lab, and it is titled Viruses as a Cystic Fibrosis Cure. And this project is building off of my last project that I did, which was just on cystic fibrosis. An introduction to cystic fibrosis. Cystic fibrosis is a serious disease that affects the lungs and digestive system, causing pancreatic insufficiency and recurrent respiratory infections. It results from a deletion-type mutation in the CFTR gene. This disease is most common in Caucasian populations and is autosomal recessive. There is currently no cure for cystic fibrosis. Treatments include medicines to help control symptoms, prevent complications, and make the condition easier to live with. Prenatal screening is also an option for parents of unborn children. This is the healthy CFTR gene. The PDF code is 5UAK, and it is pictured on the left. In a person without cystic fibrosis, the cystic fibrosis transmembrane regulator protein functions as a chloride ion channel that regulates fluid in airways and other regions. The CFTR gene is located on chromosome 7. As we can see on the left, whenever the protein is phosphorylated, the channel opens and allows ions to pass through, and this allows the cell to regulate fluids in the lungs. And this is unhealthy CFTR gene, which leads to an unhealthy CFTR protein. And the PDF code for this is 4WZ6. The most common mutation that leads to cystic fibrosis is called delta F508. This mutation involves the deletion of 3 nucleotides and causes 70% of all cystic fibrosis cases. And on the top left, I have a model of the 2 CFTR proteins on top of each other, and this region is just the regulatory domain because that's where the mutations occur, and the red parts are the mutations. The CFTR protein in cystic fibrosis cannot properly manage fluid regulation due to its structure in the regulatory domain, which leads to the channel not being able to open when the regulatory domain is phosphorylated. So basically what happens is the phosphate activates the regulatory domain that tells the channel to open, but in people with cystic fibrosis, this doesn't happen because of mutations that happen in the regulatory domain, and this leads to lots of problems. For someone diagnosed with cystic fibrosis, dealing with this disease can be extremely emotionally distressing. Patients face low life expectancy. The life expectancy is only 37 years, chronic symptoms, and decreasing bodily function that causes disability. As patients enter adulthood, their emotional well-being is known to decrease as their condition becomes more severe. Because of the disease's specific nature, there has been no success curing it. So what is the solution? For a disease this serious, it is very clear that treating the symptoms has its limits. It is necessary to tackle the issue at the genetic level using gene therapy. What is gene therapy, and could it cure cystic fibrosis? Gene therapy introduces healthy genetic material into cells in order to correct abnormalities that cause disease. Vectors are the carriers of this genetic material. They are usually modified viruses. So in this diagram, it shows a virus carrying the CFTR gene. It would infect the person through the respiratory tract, enter the lungs, infect the cells with the protein, and deliver the therapeutic genes that would in theory cure the person. Cystic fibrosis was one of the first targets of gene therapy when the practice picked up viability 2 decades ago. However, there has been no lasting success so far. Targeting the lungs is particularly tricky because our lungs are evolved to be extremely virus resistant. Even though our lungs are this way, we know that viruses like coronaviruses, influenza, adenoviruses, lentiviruses, and others do succeed in causing respiratory illness. Could we use one of these as a vector and cure cystic fibrosis? So obviously my answer to that would be yes, we probably could. And out of the viruses listed in the last slide, adenoviruses and lentiviruses are the most promising, and here's why. So both of them do really well in terms of the 4 main categories that are considered when looking at possible vectors. And these are duration of expression, targeted cells, packaging capacity, and immunogenic potential. For adenovirus vectors, it is transient, which means it doesn't integrate into the genome. And for the lentivirus, it does integrate into the genome, which makes it, in theory, a permanent solution, while the adenovirus would need to be reapplied. Targeted cells, both of them are respiratory and widespread, meaning they could infect the respiratory cells and also other cells in the body. Packaging capacity, this is how much space the vector has to hold therapeutic gene material. And for the adenovirus, it is 7.5 kb, which stands for kilobases, and for the lentivirus, it is 9.7 kilobases. The immunogenic potential means how likely is it to trigger an immune response like anaphylaxis in the patient that is given to, and obviously we don't want that to happen because that's very dangerous. And the adenovirus vector, it's considered pretty high, and for the lentivirus, it's considered relatively low. So even though both candidates are promising, lentivirus is the most promising due to its long-term expression, slightly larger packaging potential, and low probability of immune response. So putting it together, the healthy CFTR gene is approximately 189 kb in length, which greatly exceeds the 9.7 kb packaging capacity. Luckily, studies suggest that only 5 to 10% of normal CFTR gene is required to reverse disease effect. And this is because only this region of the protein is not functioning. The rest of it is okay, and we don't need to use precious space in the packaging capacity to store this information when it's already okay. We need this information, and that only accounts for 5 to 10%. So the mutated CFTR gene is in the section that codes for the regulatory domain, which is called NDB1 and is located on exon 9. So this region is the region we want to take that 5 to 10% because this region codes for this region, which is the problem. We only have 9.7 kb from exon 9, and that accounts for 5.15% of the normal CFTR gene, which does fall into the range of what is considered enough to reverse the disease effect. So yes, the lentivirus does have enough space, and it might be able to hold all the information needed to reverse the disease's effect. So summary. Cystic fibrosis is a serious disease of the transmembrane proteins involved in chloride ion transport. It is currently incurable, and that's really why I wanted to look into cures or possible cures for my final project. Healthy CFTR proteins are able to open after the NBD1 region of the protein is phosphorylated, but in people with the delta F508 mutation, this region is deformed and unable to open. Using gene therapy, this illness may be able to be cured. The ideal viral vector for the delivery of the therapeutic gene would be a modified lentivirus because of its duration of expression, packaging capacity, and low immunogenic potential. Modifying a lentivirus and packaging it with genetic material from exon 9 might be able to cure cystic fibrosis. And there's my project. So what I learned is all about viral vectors because I knew about them and I knew about their use in gene therapy, but I didn't really know why we chose the viruses that scientists choose and why they're able to be effective. It was really interesting to research all about that. And also there are promising studies using the lentivirus in humans to cure cystic fibrosis, but up until now there hasn't been a total cure, but it is looking like lentivirus might be the way to go. And now that I've done my own research into why that is and why it makes sense, I understand. Here is my links for raw data and reference publications. And that was my presentation. Thank you for watching.